Hi everyone, thank you so much for joining us once again for our Cytobank Bootcamp series. I'm Stephanie Trop. I'm a data scientist with Cytobank and Beckman Coulter Life Sciences. Today, our session will be on using a really powerful tool in Cytobank, Citrus, for automated biomarker identification. Today, we'll talk about the need for biomarker discovery in your cytometry workflow and try to understand the basic concept of classification models, an important component of how the Citrus tool works. Then, we'll see how those concepts work within Citrus and apply Citrus to analyze a mass cytometry dataset for biomarker discovery in Cytobank. As we've seen before, we really want to illustrate the context of using these tools we've been learning about within your complete data analysis workflow. Before you begin, it's really important that you properly prepare your data with good controls during data acquisition, check your compensation and apply appropriate scaling, and do pre-gating to exclude dead cells, debris, aggregates, or unstable segments of data acquisition. An optional step in your data analysis is using VisNi for dimension reduction to show you a visual landscape of your high-dimensional data. Last week, we reviewed how to use FlowSum for clustering, and today we'll use the Citrus tool, which in one step both clusters your data and compares groups of samples to identify significant differences between them. Citrus is an automated method for biomarker discovery, which is really at the heart of many of our cytometry studies. Biomarker identification can be used to understand biological mechanisms behind an outcome. For example, identifying which cell populations are associated with patients that respond successfully to immunotherapy can help us understand exactly how that therapy is working, like what cells are being targeted. Likewise, a vaccination study comparing those that did and did not generate a protective immune response can help us understand how that vaccine works. But furthermore, with the right experimental design and statistical tools, biomarkers can be used to predict outcome. For example, to help decide which treatment will be the most successful for a specific patient or stage of disease. But with high-dimensional data, it's challenging and cumbersome to use traditional analysis methods for biomarker discovery and give you confidence that you're not missing something. These problems can be alleviated with the Citrus algorithm, which is a data-driven approach to identify differences in your multidimensional cytometry data. It's automated, objective, scalable, and sensitive to detect differences. That said, Citrus is a pretty complex tool, and today we hope to introduce you to it and develop enough understanding on how to use it in your workflow for the most success. So let's start with a simple example. Living in this pandemic time, we're all engaging in different behaviors, for example, attending this webinar to avoid exposure to COVID-19 and keep ourselves healthy. A well-documented and commonly utilized method of enhancing immunity is by increasing vitamin C intake. So I currently have a lot of fruit in my kitchen. Here's a subset of 17 fruits from my entire current fruit collection. I know that citrus fruits are high in vitamin C, so I want to prioritize eating the citrus first. If we know that the fruits on the left are citrus fruits and on the right are not, can we use data about these fruits to determine what features are associated with being a citrus, and further use those features to correctly identify or classify a new piece of fruit as citrus or not citrus? By answering these two questions, we'll actually be generating correlative and predictive models. So if I knew nothing else about the fruit besides what I can see, here's some of the data that I can collect. I took notes on each fruit's appearance, including their color, size, shape, and texture. Crucial for building my model, I also know the group assignment for each sample, that is, whether the item is a citrus fruit or not. For example, I described the banana as yellow, large, banana-shaped, and smooth, and not a citrus fruit. I also had in my sample some Asian pears, apples, plums, lemons, limes, mineola, and an unripe green-colored mango. So let's examine how well each feature can be built into a model to correlate with or predict what is a citrus fruit. If we start by looking at color, I'll define here a model where citrus fruits are yellow or orange, and all the other fruits are other colors. So we can first assess the correlation between color and citrus status with a simple statistical test, like Fisher's exact test. This test shows me that my p-value is 0.34 for a correlation of citrus with yellow-orange color. If instead we use a single variable prediction model where we predict that all fruits that are yellow and orange are citrus, here's how we would guess which fruits are citrus. We correctly classify the lemons, oranges, and migniolas as citrus, but we incorrectly call the pears, apples, and bananas citrus. On the other hand, we correctly classify the green avocados and mangoes and purple plums as not citrus, and we fail to classify the lime as citrus because it's green. So we can assess the accuracy of our predictions with the model error rate. We made 5 incorrect guesses for the 17 fruits, making our model error rate 5 out of 17. For the single variable of shape, if I correlate citrus with spheres and non-citrus as other shapes, the correlation is quite good with a Fisher's p-value of 0.04, though my prediction error rate is still 5 out of 17. So we can go through and look at our size feature, the correlation and predictive value of size, which gives me a p-value of 0.34 and a model error rate of 7 out of 17. And finally, texture. If I guess that all citrus were bumpy and not citrus were smooth, the texture variable actually gives me a pretty reliable association with a p-value of 0.002 and a pretty good model error rate at 2 out of 17 for predicting citrus status. So here's a summary of all the correlations between citrus and each single feature: color, shape, size, and texture. If I look for relationships between any single variable and citrus status, I find that spherical shape and bumpy texture are each significantly associated with being a citrus. So let's say I can only eat 1 fruit per day, and I really want to make sure I maximize my vitamin C intake by eating a citrus fruit. Which feature can I use to best predict which fruit is a citrus? The best I can do is to use bumpy texture, where I'll guess incorrectly only 2 out of 17 guesses, which correspond to the 2 avocados that were in my sample. But we can improve the model accuracy by combining variables, generating a multivariate model to do better. If I combine the single best predictor, texture, with another feature, I can reduce the error rate all the way to 0. So within my sample, by combining bumpy texture with round shape, I can now correctly classify a citrus fruit 100% of the time. This is similar to how we will build predictive models with the PAM or lasso algorithms of the citrus tool on much higher dimensional data. So after I built my model, I realized I wanted to learn about what qualities are actually used to define citrus fruit. I learned that there are, in fact, non-spherical citrus fruits that exist in the world outside of my kitchen sample, like the citron and papaya shown here. My model would have excluded these elongated fruits as citrus. I made the mistake of overfitting the model, meaning it's too specific to my small sample. Fortunately, the predictive modeling algorithms in the citrus tool have ways to deal with this by favoring simpler, more broadly applicable models. So here, with my fruit classification problem, we use a simple mental algorithm to identify these simple features for a small sample. For our more complex, higher dimensional data, we'll now turn to citrus to solve these classification problems. Citrus is the tool that we'll be using for biomarker discovery with our more complex single-cell data. It's an acronym for Cluster Identification, Characterization, and Regression, and it was published in 2014. Citrus works in 2 steps to automatically identify stratifying signatures, or biomarkers, in cell subpopulations. First, it performs unsupervised clustering. Then, using group assignments specified by the user, it performs supervised statistical association to compare groups of samples. The result is features that are significantly different between groups. The features can be either abundance of a cell cluster or levels of median marker expression within a cluster. Citrus provides results in the form of lists of features, plots of clusters and their marker expression, and tables showing cluster abundance and median marker expression. Citrus is currently very popular and widely published across many different fields with both mass and fluorescent cytometry data. Just a handful of recent publications utilizing Citrus are shown here in studies of trauma, cancer, autoimmunity, cell signaling mechanisms, and infectious disease. So here's the high-level view of the steps performed by the user and by Citrus to discover significant biomarkers. First, you have to assign groups to your FCS files in Cytobank. This can be 2 or more groups that might correspond to citrus fruit versus non-citrus fruit, controls versus an experimental condition, baseline versus post-treatment, responders and non-responders, or even 3 different treatments. Along with some other basic settings, then you will run Citrus, and Citrus will perform hierarchical clustering to identify cell populations, and then compare the clusters by group. Statistical testing will result in lists of significant features. The user then takes these results and need to assess them for validity and interpret them within the context of your experiment. Then you can report your findings. Here's the high-level view of the steps performed by the user and by Citrus to discover significant biomarkers. First, you have to assign your FCS files in Cytobank into groups. This can be 2 or more groups that might correspond to citrus fruits versus non-citrus fruits, controls and experimental conditions, baseline and post-treatment, responders, non-responders, or even 3 different treatments. Along with some other basic settings, you run Citrus, and then Citrus will perform hierarchical clustering to identify cell populations, and then compare the clusters by group. Statistical testing will result in lists of significant features. The user needs to take these results and assess them, assessing the validity and interpret them within the context of your experiment. Then you can report your findings. In a little more detail, Citrus performs hierarchical clustering to identify populations. This means that you don't have to do any manual gating beyond cleaning up your data down to live single-cell events. The clusters are presented in a tree to identify both the marker expression as well as which clusters are significantly different between groups. Behind the scenes, Citrus does this by preparing your data by downsampling if needed and then combining samples. It performs agglomerative hierarchical clustering, which identifies groups of cells that are phenotypically similar. It will generate smaller and smaller clusters until reaching a % minimum cluster size, which is a user-specified setting. Then, the data are divided back into individual samples, and each cluster and sample is described by abundance and marker expression to define differences across groups. The clusters are analyzed with different statistical models selected by the user to identify which features are associated with or associated strongly enough that they can predict group or outcome. The predictive models used involve regularized regression and cross-validation to ensure that the model generated can best predict group. The significant features are plotted on box plots to compare groups, and cell events are assigned to specific clusters that you can visualize in traditional biaxial plots to confirm the findings. The generation of predictive models used by Citrus are very complex, but I hope that today we can understand a couple of important principles behind them. As we saw with the citrus fruit example, A classification model identifies important features that can distinguish groups. We saw with the fruit that the features of size and color did not distinguish citrus fruits from other fruits, but texture and shape did. Seeing the association between these individual features and citrus fruits is similar to what the citrus tool does when it builds correlative models using an algorithm called SAM. SAM results include all features that are significantly different at different false discovery rates. When we used the fruit features to predict if a fruit was a citrus or not, we generated a type of predictive model. Citrus uses two predictive models called PAM and LASSO. The predictive models can assess the effect of interactions between multiple variables. For example, how we saw that by looking at both fruit texture and shape, we had a model that best predicted if a fruit was a citrus. However, we also saw that the model with two features instead of one was overfitted to my sample. The model would be wrong in the case of the oblong citron fruits. To prevent that overfitting, PAM and LASSO use methods of regularization and cross-validation, which I didn't use in my very simple fruit model. Regularization assigns a penalty to the model for adding more features. This rewards the simplest model that can be the most broadly applicable. Cross-validation is another strategy used by PAM and LASSO in citrus to prevent overfitting. In cross-validation, a portion of the data is omitted for model training or building, then the model is tested on that left-out data. Specifically, citrus uses k-fold cross-validation, which is important to understand for knowing how to set up your citrus run. Your samples will be divided into k-equal groups, where you have to decide how many groups or folds you want to use. For example, if you're comparing a healthy group of 25 samples and a patient group of 25 samples, you have 50 samples total, and choose a cross-validation fold of 5. Your model will be built on 40 samples and tested on the leftover 10 samples, picking if each of those 10 samples came from a healthy or patient subject. The number of times the model guessed incorrectly is called the test error rate. The prediction is then repeated 5 times for each cross-validation fold, and the final cross-validation error rate is the average of the 5 test error rates. You need a model that can get this error rate sufficiently low for your application. If the consequences are high for a wrong guess, you need the error rate to be 0. If the consequences are low for a wrong guess, and you're more interested in exploration, it might be okay if the error rate is not close to 0. For example, if I ate a mango instead of a mandarin because my fruit model was wrong, it wouldn't really have been that serious a problem. So this cross-validation is performed in conjunction with regularization to evaluate multiple models. The best model that results from citrus is the simplest model that falls within your acceptable error constraints. So far, citrus has done all the heavy lifting. But with the resulting models that citrus generates, you have to review the results. You need to assess if the results meet your needs for statistical significance, ensuring that you have enough samples in each group and that your cross-validation error rate is sufficiently low and within your false discovery rate. Then you need to phenotype the significant cell clusters and understand the pattern of how they behave in each group. You can do this by looking at histograms of marker expression and box plots of cluster abundance provided by citrus, as well as exporting the significant clusters into a new Cytobank experiment. Finally, you have to decide if knowing which clusters are significant and in which group they are elevated or decreased, if that's consistent with your previous knowledge about the biological system. We'll finally look at some real single-cell data using citrus. We'll be using the same dataset that Chen-Jun used to demonstrate Disney plots last week. This is a mass cytometry study of PBMCs from pediatric cases of chikungunya virus infection in Nicaragua. We'll be comparing samples collected during the acute and convalescent infection and use citrus to identify biomarkers of acute infection. We'll be looking at 10 subjects per time point. So referring back to our workflow, we need to first define the groups for comparison, which are convalescent and acute time points. Then we'll set up our citrus run and interpret the results. So let's take a look into Cytobank. We'll set up a citrus run on abundance, and then we'll look at some results from the predictive model PAM. So here we are in Cytobank in a Visney experiment. You can see this experiment has 20 FCS files with 10 files collected at the convalescent time point and 10 files from the acute time point. And we'll be starting from a Visney result that was run on CD45 positive cells. So to create a new citrus analysis, we'll come up here to the navigation bar, click Advanced Analyses, and create a new citrus analysis. We'll give it a name. And this will pull up our settings page. So we'll set up a run, while on the left you can read about these settings in more detail. First, we'll choose our starting population. If this is my first run in an experiment, I think I want to start pretty broad, so I'll start with CD45 positive cells. If you want to look at more specific cell subsets, you can select a smaller population. File grouping. This is where I'll choose which group to assign each file into. So I'll click this Add File Group button, give my groups names, in this case acute and con for convalescent. These also match strings that are in the FCS file names, so that made it really easy for Cytobank to search and automatically group specific FCS files into the corresponding group name. So I can see that all my files have been properly assigned to groups. I can also see here how many events are in each of my files, as well as how many events are in the selected CD45 positive population. Next, I need to decide which channels to do clustering on. I want to start pretty broad here. I'll choose all of the major phenotyping markers, and those were all the same markers that I included in the Visney run, so it's really easy for me to find them. Uncheck any channels that I don't want here, and that leaves me with 17 channels, my more important phenotyping markers. Then we choose which of 1, 2, or 3 association models we want to use. I recommend choosing all 3. But SAM is the correlative method, PAM and lasso are the predictive methods. The only reason that you couldn't use one of these would be if you had more than 2 groups, in which case you could not select lasso. Then we will choose to run in abundance or median mode. In abundance mode, we'll be comparing the abundance of each cell cluster across groups, or in median mode, we'll be comparing median marker expression of selected markers within clusters across groups. So if we choose abundance, that's the only setting we need to choose. If we switch to median mode, another window will pop up where we can choose which channels we want to use for statistics, which channels we want to be used for comparison. And these channels, in that case, you probably want to be different than the channels that you select up here for clustering. Today we'll use abundance. Then you need to choose whether to use equal sampling or the maximum number of events per file. In abundance mode, you're only allowed to use equal sampling. For additional settings, you could choose the number of events to sample per file. If you want to choose the maximum that you have, I'll just add another digit there, and that maximum will be automatically calculated. Then you could choose the minimum cluster size. This is a percentage of total events in your sample. If you expect that a very large population is important, you can leave this default of 5%. If you think you need to hone in on much smaller populations to find the significant differences to find your biomarkers, you can lower this minimum cluster size. The smaller you make that minimum cluster size, the longer it'll take your run, the longer the clustering step will take. Next, you can choose the number of cross-validation folds, like we talked about, the number of groups that you want your samples to be divided into for cross-validation purposes. The higher number you choose here, the more robust your results will be, the longer it will take as well, because every fold you have is another replicate of the step of model building and testing. 5 is the default. More stringent, you can go higher. And then we have false discovery rate. Decreasing this value will give you fewer significant results with higher confidence and vice versa. Finally, you can choose whether or not to apply a z-score normalization across your scales, and you could choose a light or a dark plot theme for your plots. So when I'm happy with my settings, I can run the citrus analysis. So this will take some time, and how long it takes will depend on the number of events you have, the number of clustering channels you have, and the cross-validation folds, as well as the minimum cluster size. So instead of waiting for this to complete, we'll look at results that I already ran with these same settings. So if I click here on Advanced Analyses, in addition to being able to create a new analysis, I can also see all of my other runs. So I will click on a run that has matching results to this one. When your analysis is complete, you can always revisit your settings page. You'll see this analysis complete message. You can download the results, and you can also export any clusters that you're interested in into a new experiment. So let's take a look at what's included in these downloaded files. When your run is complete, you'll download a pretty large package of results. They always stay attached to your Cytobank experiment, so you don't have to worry about where you store this downloaded file. The files will include all the information about the settings for your run, plots that show the marker expression of each cluster, and separate folders of results for each association model that you ran. That's SAM, PAM, and Lasso. Within each association mode, you'll find the results for up to 3 different models representing the significant features under different statistical thresholds. For SAM, these thresholds correspond to false discovery rates of 1, 5, or 10%. For PAM and Lasso, they apply to different cross-validation models that we'll look at in a minute. For each model and statistical threshold, there are plots of the cluster trees, histograms of marker expression, and box plots included. The predictive models will have model error rate plots to assess the quality of the model. And each model also has tables of cluster abundance or median. When you download the results for a predictive model, PAM or Lasso, the first thing you need to do is to look at the model error rate plot to assess the validity of your model. As we talked about, Citrus uses regularization and cross-validation to evaluate models with different features to find the best model for predicting group outcome. We'll look at this model error rate plot to see the error rate of each model. First, this red line plots the cross-validation error rate as a function of the number of model features and an inverse of the regularization threshold. So as we move to the right and increase the number of features or cell clusters in the model, we expect the model to be a better predictor of outcome, which is what we see as the cross-validation error rate decreases. At the same time, as the number of features increases, the false discovery rate, or the chance of finding false features that are false positives, will increase. So up to 3 different models are built along this continuum. The model called CV-min is the model that has the lowest cross-validation error rate. The CV1SE model is the model with the fewest number of features at the lowest cross-validation error rate plus 1 standard error. This is often considered to be the best model to predict new data. The third model is the CV-FDR constrained model, which has the highest number of features while still staying below the false discovery rate. So we can take a look at the significant features in each of these models. You can see that the CV-min and CV1SE model each have only 1 feature, while the FDR constrained model has about 23 features. These models were all generated using the same settings we just set up in Cytobank, using the major phenotyping markers, a 5% minimum cluster size, and a 1% false discovery rate. These 3 corresponding trees show us where the significant features are in each model. You can see that they all contain one specific cluster, number 357869, But additionally, the FDR constraint model has several more features that fall into 3 separate branches on the tree. To interpret the citrus tree, you can see that each cluster has a number, which is its identifier. The center cluster has 2 arrows pointing away from it. This cluster represents all of the events. In generating the clusters, 2 children are created for every parent until reaching the minimum cluster size threshold set by the user. As you go down each tree lineage, the clusters get smaller and smaller. All children are also contained in the parent cluster. This means that there is redundancy in cell events in the clusters. Cells can be assigned to multiple clusters. This allows the statistical comparison of different populations, both big and small. Next, we can use plots of marker expression to phenotype the significant clusters. By scrolling through the plots, I can survey the landscape, and I identified the major populations associated with each branch of the tree. I see that the significant cluster is the parent-most cluster of a branch of CD14 positive cells. I can further identify the phenotype of this cluster with the downloaded histograms of marker expression of the significant cluster in red as compared to the rest of the cell events in blue. I can see that this cluster has mid CD4 expression, high CD86, high CD14, CD54, and CD11b. So these are some sort of monocyte. And we saw it's a really big population because it's very close to the center of the tree. So this probably represents a mixed population of monocytes. The downloaded results also come with box plots of the abundance of this cluster across all files in each group. You want to make sure the difference in abundance is biologically meaningful. In this case, we're looking at a very large population of around 40% of the cells in the acute samples versus around 10% in the convalescent samples, a very convincing difference. After phenotyping the significant features, you should look at them in the context of what you know, of visualizations that you're confident in, like traditional biaxial plots, and in the biology that you know. To do that, you can export clusters of interest into a new experiment. Here's our citrus results page. So from here I can click Export Clusters to New Experiment. I'll select which cluster or clusters I want to export, along with all of the original FCS files to include. So the cluster files will only have the events that were in that cluster, and the original files will have all of the events. I'll start export, and this will make a new experiment, which will forever be linked to this citrus settings page. I already exported this cluster previously, so I'll open up that result experiment. So up here, this shows me the origins of this experiment. It's a citrus experiment. I can see there's citrus results from here. I can see which VISD result this came from. And here's where I am on my experiment tree. If I scroll down to look at all the FCS files in my experiment, I now have 40 FCS files. I started with 20 individual files, and then I exported the significant cluster for each file. So now, if you look at these top 2 rows, the top file is my original file, and the second file is only the events in cluster 357869. So now I've doubled my files. I can get other useful information here as well. If I look to the right, I see the number of events in each of these files. So I'll show you a couple of possible layouts for the working illustration to give you some more information on how you can interact with these data. I can overlay this significant cluster back onto my VISD map so I can see really clear changes by time point at acute and convalescent infection by looking at the difference in density in this cluster. If I want to use visualizations to look at the difference in the number of events in this cluster across time points, I can make a heat map of the number of events. I can do this by selecting a heat map plot type and coming down here, choosing my statistic of event counts. In this case, I've chosen raw, meaning the raw event counts will be shown, but I could also set this up as a comparison, say fold, to show the difference in time points, acute and convalescent, across each individual subject. I could also dive deeper into the phenotype of this cluster as compared to all the events in the sample. Here, I have set up histograms showing expression of every marker in my citrus analysis of this cluster versus all the events, looking at top at the acute samples and the bottom the convalescent samples. So you can set up your working illustration in any number of ways to show you about the phenotype of these clusters, or the size of these clusters, or the comparisons between groups. So to wrap up, after our citrus run was complete, we had to evaluate our results to make sure that they were valid and they made sense in the context of our experiment. We did that by looking at the model error rate plot. We took a look at the box plots of cluster abundance across groups to make sure that our effect size was biologically relevant and consistent across samples. We phenotyped our significant clusters by looking at marker expression and made sure that they made sense considering the biology, in this case of viral infection. And altogether, once we checked all those parts of the results, I think citrus showed us a convincingly significant expansion of CD14 positive monocytes during acute chikungunya infection. And here, we used just very broad populations looking at the most important major immune phenotyping markers. This is a great starting point, a jumping off point for maybe honing in more on this population. We could do that by doing things like including additional markers in our analysis, in our clustering, maybe running citrus on only this large CD14 positive population that we identified, or maybe using citrus in median mode to look for significant changes in marker expression. Another thing we can do is look at the other models that came with our results here. So we looked at the one significant cluster that citrus identified using only the most conservative model with the fewest features, that CV1SE model. We could also take a look at the FDR constrained model to discover additional significant clusters. So in that model, there were 23 significant clusters, and they fell into 3 groups of distinct populations. So we could choose a couple of different strategies. Maybe if we wanted to look at the larger populations, we could look at the parents of each group. Or if we wanted to look at the most pure, the smallest populations, we could look at the smallest children of each of these 3 significant branches. In our short session together today, I've shown you how citrus is a very powerful data-driven approach to automatically identifying stratifying subpopulations or biomarkers in your high-dimensional data. It's very customizable to suit your data, your scientific question, and your statistical needs. The statistical power of citrus comes from being able to set a false discovery rate and the use of regularization and cross-validation to ensure the best predictive model is being generated. You can evaluate the models by looking at the model error rate plot. And most importantly, I think, is that it's easy to integrate citrus into the rest of your workflow, easily running citrus directly from your VISNI result and then exporting significant clusters into a new experiment for further analysis. Hope I've given you lots of ideas on how citrus might apply nicely to your own data. So here are some resources to get started or continue with using Cytobank. You could sign up for a free 30-day trial by visiting premium.cytobank.org. You can visit our website and our blog at blog.cytobank.org for educational articles. Our support site is support.cytobank.org with lots of support documentation. That's also where you can submit a request for technical support. We also offer training and consulting services, including consultations with our scientific services team on single-cell data analysis projects. And you can inquire about those by visiting support.cytobank.org as well. Thank you so much again for joining us today, and we'll take any questions you have now.
Thank you, Steph, so much. That was really great. Great use of all of your citrus fruits, especially in this time. So thank you for that awesome comparison.
Thank you. I was really inspired.
Yeah, that's how I learn, through metaphors. All right. So what we're going to do right now, thank you all for attending first and foremost. I'm going to introduce all of our panelists. So as you heard, Dr. Steph Trop is our data scientist with Cytobank, and she's going to be tackling some of the questions along with Chin Jun, who is our another data scientist on the West Coast. We also have our data scientist, sorry, I apologize, data scientist, Steph is the data scientist, Chin Jun is the application scientist. And we have Chi-Hao, who is also a data scientist with Cytobank, and Stephanie, oh my goodness, Sarah Schuett, who's our sales for the East Coast, and also a new sales and a new application scientist addition to our team, Jason. Okay, so without further ado, we're going to go delve into some of these questions. The first question is there any flow demo data in Cytobank to use for these applications? And I'm going to direct this to Steph.
Sure, yeah. So we have a public experiment available on all Cytobank servers. I think it's called Citrus Demo, but it's good data on B-cell receptor cross-linking. And if you log into your Cytobank account, and you can access this whether you already have a Cytobank subscription or you're on the free trial, and click Cytobank curated experiments. It is this one here. It says Citrus Demo. And this experiment in particular might be really useful for illustrating how to use Citrus in median mode.
Perfect. Thank you. All right, from an anonymous attendee, how many samples per comparison group are needed to run the Citrus analysis? And I'm going to get Chin Jun to answer this question, please.
Sure, I'll be happy to take that question. So for the algorithm, the minimum number of samples to take is 3. However, in order to have the statistical power, we recommend to have at least 8 samples per group to run Citrus so that the results is statistically solid. So if you have too few samples, the result might be doubtful and not convincing. So 8 samples minimum to run Citrus.
Thank you, Chin Jun. All right, I'm going to direct this question to Chi-Hao. Can Citrus be used for non-facts or non-site-of data, even when that data are not numerically, are not necessarily all numerical values or numerical values that do not have the same scale or range?
And thanks, Alina. And unfortunately, like Citrus cannot be run with non-numerical data. And so if you have, like, for example, you have like single cell RMAC data or like single cell like SIC data set, and the data are all numerical values. And yes, you can run Citrus with those kind of data set. But again, like, you should not use Citrus to analyzing your BARC RMAC data, because Citrus is the tools to analyzing single cell data set. It's like you cannot use it to analyze a like BARC sample, like those kind of data. And about data scaling, yes, if you're analyzing some non-cytometry data, we highly recommend you turn on the data scaling option.
Perfect. Thank you, Chi-Hao. Okay, another question. This is basically a clarification question. So Citrus is a similar model. Is Citrus a similar model to TISNI? But instead it identifies population in a little more enhanced or different way, such that it gives you additional or different information about the population of interest. Chi-Hao, is that the case?
Not really. Actually, Citrus is different from TISNI or VISNI, and like Citrus had, like Stephanie mentioned in the talk, Citrus has like 2 main components. The first one is like clustering, and the second part is statistical testing. So you can use TISNI to do the clustering part, but like TISNI cannot do the statistical testing for you. And Citrus is really like end-to-end solution for you to identify biomarker. It will automatically identify cell clusters using clustering algorithm, and then do statistical testing on those identified cell clusters to find the statistical significant biomarker for you.
Okay, perfect. Okay, so what is the difference between Citrus and doing a non-parametric test on flow sum metaclusters? in cases of two groups, healthy and diseased, such that you look at the metacluster, such that the difference between the two metaclusters is significant between the two conditions.
And actually, I want to remind everyone, Citrus has 3 different like statistical testing methods: SAM, PEN, and LASSO. And I think like the flow sum plus non-parametric test approach is similar to the SAM method. Because like again, like the non-parametric test will test the variables like one by one. And so, but like if you run like PEN or LASSO, like PEN and LASSO, they are like multivariate approach. That means when they build a model and when the algorithm build a model, you will like evaluate more than one variables at the same time, and to get better prediction accuracy. And then later the algorithm will base on the prediction model to inform which biomarker or cluster are statistically significant. And I guess in short, yes, you can do that. But like Citrus has 2 other algorithms which we think on certain cases is much better than the regular SAM method or than the flow sum plus non-parametric testing method. But I can see the flow sum plus non-parametric testing method approach is very flexible, and you can easily like kind of construct whatever test you want to do. And I think it's like, yeah, if you're familiar with doing non-parametric tests, and yes, you can do it for that.
Okay, thank you so much. All right, is it necessary to create a VISNI analysis prior to Citrus? And I'm gonna get Stephanie to answer this question.
Thanks, that's a good question because that's why I showed it here, but it's not necessary at all to run a VISNI first. I like to do it, and we recommend that if you have the time and the right number of events, because it gives you a really nice simple visualization, which I hope that you saw when I overlaid that significant cluster on the VISNI map. It was really easy to see where those events were when looking at all of the events in my sample and compare them side by side visually. So it's not necessary, but I think it gives you a very helpful, clear visualization.
Thank you, Steph. Since we're on the same topic, I'm just going to ask you another question. And does it make a difference to use separate FCS files for each patient, or do you want to concatenate these for the whole group?
Yeah, it definitely makes a difference. And where Citrus thrives is in dealing with groups of multiple samples. The things that we talked about, like cross-validation and predicting outcome, that can only be done on groups of samples. So the statistical power of Citrus comes from having those replicates in your groups.
Okay, thank you. Jihao, could you answer this one? How would one statistically adjust for unequal group sizes?
Yes. I guess I want to clarify that there is no statistical like method that will allow you to adjust for adjust for the p-value after you're doing some kind of testing with an unequal group experiment. But what you can do is like you pick the right statistical method to do that. Like for example, if you really have an unequal group size, you probably should go to use some non-parametric methods to do the statistical testing rather than using the parametric methods, because parametric method often assume the background distribution is a normal distribution, and you need to have like an equal balanced group size in your experiment. And so in those cases, like I guess the solution is like pick the right statistical test to do that.
Thank you very much. Okay, we have a question that's more specific, so we're going to try to tackle that one. All right. Laura says, I've tried to use the LASSO model to predict which samples belong to a certain patient groups. I had 2 groups in total. A number of clusters were identified as being significantly different. However, when I inspected the cluster abundance box plots, they didn't visually appear to be different. Do you know why might this be the case?
Yes, this is a great question. Thanks, Alina. And I guess the first thing I suggest that you should look at what is the cross validation error rate of the lasso model you get, and is that error rate like really high or acceptable? And because it is possible that you can get a model, but the model has very high error rate, and for those kind of model, it will not be very surprised to see some like biomarker clusters that are not significant, like kind of visually different. And the other thing I would like to mention is that I don't know how many samples per group do you have, but if you really have a lot of samples within each group, and it is possible that like visually you don't see a big difference, but like statistically it's still significant, because the statistical method will kind of like calculate a ratio, like kind of a within group variance, and divided that by the between group variance. So like if you have within group like a large within group variabilities, and even though the between group variability is small, and like statistical testing can still identify those kind of a cluster as a significant cluster.
That's great. Thank you very much, Chihao. All right. What is the minimum number of events sampled per file for citrus? And is number below 5,000 events reliable? Junjun, do you want to tackle this one?
Sure, I'll be happy to take that one. The minimum number of events per file, it will really depend on what is the minimum number or the rare event of the subsets you are looking for the citrus. So if you are working on rare subset that's under 5%, there is a setting in citrus where you can define what is the minimum number of clusters you would like to identify for that one. So it's really, is the number below 5,000, is that per file reliable? That again, it's pretty much the same question of, what is the rare population you're looking for? We want to back calculate, first look at your rare population, then back calculate what will be the minimum number of input you would like to have for the citrus in order to identify your rare population. And we will be happy to share a publication on that for you as well. So feel free to write into the support. We will be happy to share the calculation for you.
Thank you, Chinjun. Okay, a question from Vivek. Can I choose which FDR method Citrus uses? Chihao, would you mind answering this?
Yeah, sure. And yeah, I know there are many different FDR correction methods available. Unfortunately, in Cytobank, yeah, you cannot choose which one do you prefer. And actually, I believe in Cytobank, we are using the Benjamin Hagberg method, which is the most stringent method. And yeah, that's what we have in the Cytobank. Perfect.
Thank you very much. Okay, Steph, a question for you. Can you do group statistical analysis on clusters, and which data to use for it? Is it abundance?
Okay, so if you want to look in more detail at the results of the group-wise comparisons, you can go to your downloaded files, which I have here, and you would download, take a look at the abundance information. So you'll have up to 3 files here that correspond to the different cross-validation models that we talked about. And for instance, if we open up this one, which should be the model that had 1 feature, yeah, you'll have a spreadsheet with a list of each of your files and the abundance of that cluster. So you can compare, yes, cluster abundance if you're running in abundance mode. If you're running in median mode, then you'll have a table that has the median marker expression for each cluster, for each file, and you can compare those.
Thank you, Steph. Okay, going along. Can you please explain the citrus overlaid on Visney map again? And Steph, do you want to try that one again?
Sure, yeah. So as we already talked about with someone else's question, this is something that you can do to just get a really simple visual overview of your data. So the way that I got there was to export a cluster that I was interested in into a new experiment. And I have that here. We'll take a look at how I set this up. So the first thing I wanted to do was tag my cluster files. You can go to sample tags for that, and you can put this into any sample tag. I put it in sample type. So what I did was I typed in this cluster number to identify all the cluster files and assign them to this tag, and then assign the rest of the files to another tag. And by doing this, I can nicely overlay them in the working illustration.
Okay, perfect. Thank you very much. Okay, we have another question, a really good one. What are the limits on the number of channels and features that can be analyzed in Citrus? This is with regards to AbSeq or CiteSeq experiments with more than 100 features, or even with respect to the RNA-seq data. And Jiho, do you want to answer this one?
Yeah, sure. And there's no like kind of a official limit for number of events or features you can you can run with Citrus. But like, I mean, Cytobank has a limit. Like if you are a premium user, you only can import less than 200 channels of data into Cytobank. And if you are an enterprise customer, you can import up to 818 channels into Cytobank, and you can like basically you can run Citrus analysis with a data set that has like 800 channels, and that would work for you. But one consideration is that Citrus used the hierarchical clustering to identify cell cluster. Hierarchical clustering is a very classical, like kind of a golden standard method, but unfortunately, it is not very computational efficient. And then, so that means if you have a large data set, for example, you have like, I don't know, in total, like 2 million cell events, and you can run like hierarchical clustering can clustering cell events with 2 million events, but it will be very, very slow. And also like for example, you have like single cell RNA-seq data, like you usually have a lot of like channels or features, but you only have a limited number of cell events. You probably will have less than like 10,000 events. You probably only have like maybe like 8,000 analysis cells, but you will have like probably like 500 or 600 genes or most variable genes that you want to look at. In these cases, yes, you can run Citrus with that kind of a single cell RNA-seq data.
Perfect. Thank you so much. I'm just going to take one last question, and this is the simple one. Is there a cap in terms of the number of total events one can run using Citrus? And I'm going to get Steph to answer this.
Actually, there is not, but with increasing number of events, the algorithm will become more likely to fail. And I think that's really a combination of events and channels. So I don't have a black and white answer for that. But if Chihao or Chen-Jun have a more specific answer, please step in.
Oh, that's perfect. Yeah, that's exactly how it works in Cytobank. You can always try to see what's the next number you can input. Yeah.
Excellent. Thank you, panelists, for all your excellent tackling on the questions. And thank you all attendees for joining us yet again for another Cytobank Bootcamp session. Don't forget, we've got one more session to go this Thursday coming up at noon on the East Coast and 9 a.m. on the West Coast. So please join us for one last Cytobank Bootcamp. Until then, stay healthy and stay safe. And we look forward to seeing you again. Bye, everybody.
Thank you, everybody.
Thank you.